Yesterday we have discussed about the N-Glycosylation path, N-Glycosylation in the Golgi for the protein that are destined for either at the plasma membrane or at the proteins that are secreted outside the cell, that is intercellular proteins. And today we will discuss about the N-Glycosylation of the proteins that are destined for lysosome. There could be reason that if the proteins is not folded properly, then if the N-Glycosylation is not done properly in the Golgi, and the enzyme in the Golgi determines that it is not in its native form, then it will provide a signature to the glycan structure. Then that will be detected. Then that signal will direct it towards the lysosome for degradation. So we will understand what that signal is and how that signal is incorporated in this glycan. Okay, so as we know that when the protein leaves endoplasmic reticulum, folded proteins, it has 2 N-acetyl glucosamine and 8 mannose structure. And so for lysosomal degradation signal, it occurs in the cis part of the Golgi. Golgi has 3 major sections. Golgi has 3 major sections: cis, medial, and trans. So this lysosomal degradation signal is been provided to the proteins in the cis itself. So what it does is the protein that are destined for lysosome will get attached by phosphorylated N-acetyl glucosamine. So I am denoting phosphorylated like this, N-acetyl glucosamine. After that, what happens is, and this phosphorylated is been done at a specific location of mannose. It depends on the structure of the proteins and how the enzyme interact with the proteins. After that, what happens is this N-acetyl glucosamine is removed. So now the glycan structure is only phosphorylated at a specific position. So now this glycan structure is the signal for the protein that it has to be destined towards lysosome. So the enzyme in the further part, in the trans or in the TLC region, it is trans network we will discuss about that later. After the trans, there is a network that will determine whether the protein should be go for go to plasma membrane, or it should secrete outside the cell, or it is destined towards lysosome. So at that part, this signal, this glycan structure which phosphorylated one mannose will destine towards lysosome. As we have learned in the previous videos that there is another kind of signature happened, that is in the primary chain of amino acid, where the initial few amino acid sequence act as a signal where the proteins need to be translated or secreted. For example, in the endoplasmic reticulum, when the protein starts translation in the cytosol, as soon as it's formed its signal sequence, then the ribosome will detect that signal sequence and then stop translation in the cytosol and move that ribosome machinery, and bind and dock it with the endoplasmic reticulum, and with the help of translocon it will start translating the proteins that is destined for endoplasmic reticulum inside the ER lumen. And very general in general term, if one needs to determine whether the proteins, what is the signature for the proteins that are destined in the ER, that signature is called KDEL sequence. So any protein that has this sequence, it has been considered generally that it's these proteins resides in endoplasmic reticulum. So now we have understood that how the N-Glycosylation occurs in the Golgi for the proteins that are destined for plasma membrane and secreted outside the cell, and the proteins that are destined towards lysosome. So we will discuss further next or in some video about O-glycosylation. Okay, that's all for today. Thank you.